Great, thanks. This might be shorter than 30 minutes because it's a very data light deck, but I'll try to do my best. So hi, I'm Stefan Lukianovl. I'm the founder and CEO of Salve Therapeutics, and our motto is that we build viral biologics using computational tools. So the problem that we're trying to address is that millions of patients suffer from incurable, inherited, or acquired genetic diseases, infectious microbes, and tissue injuries that demand new precision medicine solutions. And as you can see on the right-hand side, these cost the US healthcare system quite a bit. They affect about 25 to 30 million people. The pediatric rare disease economic burden is almost $1 trillion in just 2019 alone. The healthcare cost per patient is quite high, as are genetic tests that are meant to help diagnose these types of diseases. And they represent a high percentage of hospital discharges and healthcare claim costs. So they definitely need some new solutions to help address these concerns. Our idea is to bring tech to designing these medicines to make them better so that patients can lead healthier lives. And the product we're designing first to do that is called VirCAD, short for Virus Computer Aided Design. And it's an HPC AI-capable bioCAD platform similar to other CAD platforms to bioengineer new viral therapeutics or others via the following four functions. The first is bioinformatics, where the first design layer is basically where you create a vector map by mining online genomic databases for the components. Those are then translated up to another design layer, which is the structural modeling layer, where you construct novel particles from those components to shape a viral morphology, a capsid morphology. Third is molecular simulation. You test how the viral particles would access, bind, transduce, and infect the host tissue of interest. So create a virtual tissue space based on physiological data and known physical chemical principles to test how your designed virus would work for your diseased tissue of interest. And finally, all this is accelerated by high-performance computers, so engineering-type workstations, parallel GPU units that can also accomplish the ML and AI capabilities that you need for running modeling simulation like this. And this is basically bringing virtual prototyping to biotech. So our value proposition is that VirCAD is the first bioCAD platform for viral bioengineering to enable researchers to explore and mine new data for drugs to accelerate hit to lead time and expand company pipelines through rational biologic design. This is our technology overview. It's the schematic of how the software will work. We like to call it a virtual Lego kit for viral biodesign. The input data comes from viral discovery from patient sources or natural sources. As we all know, these are deposited in online databases in different file formats. These have an open API that you can access via Java connections to your desktop app. This is VirCAD itself in the gray box, and these are the three design layers that I was mentioning. First, the genome editor, where you create the vector map, the modeling track, where you create the capsid morphology, and the simulation track, where you run the mechanistic simulation of your drug of interest. Again, this all runs on HPC workstations. It can be made cloud accessible, but the customer base tends to prefer on-prem solutions, being a bit more secretive. And we hope that through this process, you can iterate and get from hit to lead faster by predicting what structures can bind to your disease tissues of interest. And this will lead to new viral entities with applications to cell and gene therapy, cancer, antibiotics, regenerative medicine, autoimmune vaccines, you name it. And the nice part is that the follow-up is already well established. So the techniques that we use for biochemistry and biophysics, as well as cell and animals, are quite established for viral type assays. And if you get from hit to lead faster, that reduces your wet lab costs as well, because you have less to test in the end through that type of validation. So why now? First is high-performance computers and other techniques are powerful enough to model and simulate biological processes. This wasn't always the case until GPUs and CPUs really reached the stage they're at and started acting in parallel. The second is that multiple complex, rare, and orphan diseases remain untreated or lack adequate treatment options. And these are very desperate patient populations that often get overlooked by big pharma. So we think that a tech solution can bring down drug prices through virtual prototyping and finally meet their needs. The third is that biologic pipelines are generally drying, not dying, due to a lack of research tools for time and cost-effective discovery and development. That's, we think, because they don't really have an engineering mindset when it comes to designing new biologics. They usually have a genetic mindset or a molecular biology mindset where it involves screens and sort of like a random test that isn't really efficient for rational design. The market landscape is quite lucrative. So the total available market for all the disease categories that I mentioned on a previous slide that can be treated with viral biologics is well over 300 billion. If we just reduce that to cell and gene therapies, including cancer, that goes down to 100 billion, still quite a bit. And then finally, if you just target gene therapies, that's 40 billion. So, excuse me, we think that having these types of drugs can present companies with quite a bit of market share. These are a little bit dated. We hope to have an alpha by sometime early this year, and then do corporate beta testing by 2024, and then have a market entry for VirCAD by 2025. So this would be the first real CAD style software for biology. And if we capture just 1% of the cell and gene therapy market, that's enough to run a drug company just by itself. Our competition is pretty light, and I don't mean to say this out of any hubris. There really, to our knowledge, is no other bioCAD platform available on the market. What researchers usually use for cell and gene therapy companies is a host of their favorite program or app or website to accomplish whatever research task they happen to be assigned at that time. So the most popular that we all probably know of are Schrodinger's Pymol. Vector NTI has been discontinued, but it's still used by some people. MATLAB is obviously very popular, as is SnapGene. And then Dassault Systems' BioVia is, I'd say, the closest that we come to true molecular dynamic MOA simulations. But it's still, you can't design a vector map with BioVia, and you can't run a simulation with Pymol as well as BioVia. So the point is that the workflow is scattered between these different software suites, and therefore you can't get a true engineering CAD style workflow to truly virtual prototype new biologics for rare indications. This is our ask. It's not really relevant to this talk, so I'll skip it. But our development timeline is as follows. We just got accepted into an incubator in Los Angeles called Braid Theory out of the Port of LA that specializes in synthetic biology. So it's really well oriented towards what we want to accomplish. The first major pitch we're doing with them is the Startup Grind Global Conference in the Bay Area next month. We hope that we'll get some money with that to file a non-provisional. Currently, we have a provisional patent on the design. We also just submitted a phase 1 NSFS TTR with a collaborator at Children's Hospital Los Angeles for non-dilutive funding to be applied to the design development of VirCAD. So hopefully we'll hear back on that in a few months. We also have an idea of acquiring Vector NTI because it's been discontinued to work into an MVP. So that would represent the genome editor layer with heavy modifications to make it work better than it did. That we would then build the modeling and simulation layers on top of. We could also do it ourselves, but something about repurposing Vector NTI just sounds kind of funny and kind of interesting. And then finally, we hope to complete the alpha. This is a little bit in contradiction to a previous slide, but complete the alpha and then start doing corporate beta testing with potential customers by 2024 to basically develop this entirely with resources from the companies we'd sell it to via B2B SaaS. This is a pretty interesting way to approach biology, and it can lead to some cool financial projections. So if this is how engineering evolved in other industries, so electronics, mechanical, chemical, etc. And what you could envision is, big companies or little startups putting together to develop really their own CAD platforms for their own indications or preferred biologics. The CAD market, aside from biology, just from SolidWorks, from Autodesk, from DraftSight, is well over 10 billion. So this is a very marketable type of software. And then if you consider how big the biologic market is, it's going to reach about 700 billion by 2030. Combined, that leads to a fertile field, we think, for CAD-mediated biologic development in biotech and biopharma. And these are just sort of our company metrics, but I think what we should focus on is this sort of software approach to biologic design could be really useful for not just our company, but really any company that wants to get into it. This is our corporate team as it stands right now. We all met virtually during workshops and pitches and competitions, and we've actually never met in person, but that actually indicates our strength as a resilient unit that's been able to push this forward, really with no funding, totally bootstrapped, while still having day jobs, since I'd say about 2021, really. And luckily, the idea has had so much kind of merit that we've been able to acquire each other, I suppose, but also some interesting advisors. One is the collaborator at Children's Hospital LA that I mentioned. We have some contractors who specialize in healthcare IT, but they also have AI and machine learning expertise, so they can help with the more nuanced design aspects of the program. And finally, these are some of the incubators and startup connections that we have with other corporations that are helping us along the way. I think that's it. I'm sorry if I'm stopping early, but thanks very much, and let me know if you have any questions.